Hello everyone, I am Sachlin, and by the end of this presentation I hope that I would be able to explain or tell you the story of RegenCOV, the almost vaccine. Well, let's start with this very interesting question. Now, I come from a third world country, India. We do not have a lot of information about Pokemon, but let's say you're having this Pokemon fight and somebody pulls out a Charizard. I have as much information about Pokemon as much as about biology, which is not a lot. So what would you do? Charizard is actually a fire and a dragon type Pokemon. What you do is you would pull something which is either a water or an electricity type, which is exactly what RegenCOV tries to do. It basically tries to pick two different antibodies that work together with the receptor binding domain of the spike protein. Well, we'll come back to this part, but let's start from just the structure of the SARS-CoV virus. Looking at this, I know it's a bit small, but also have to fit it inside this box. The pink part right here is a spike protein. A spike protein, something right here, is something that connects to the ACE2 receptor, which then basically pulls in the whole virus inside the host cell. So your spike protein basically is one part of the virus, and then the receptor binding protein is a part of the spike protein right there. So RegenCOV actually, when I say the almost vaccine, because it was used for both treatment and post-exposure. So a vaccine is something that is used before, but it is not a cure because it is not something that cures COVID-19, therefore the term the almost vaccine. Well, let's get to the development part. So there's three phases usually, the animal testing. This is by the FDA. Phase 1, phase 2, phase 3, and then the emergency use authorization. And let's get started with how RegenCOV is actually made, or how it was actually conceptualized. So I would ask if Dr. Dolan would just help me demonstrate. Well, I would ask for 2 more volunteers shortly. But so let's say she is the host cell, and I'm the SARS-CoV, and this is the receptor. These two, actually, my 2 hands, are the receptor binding proteins, or the domain, I'm sorry, the receptor binding domains. So how do I capture a host cell is basically with both the receptor binding domains, I'm able to make hostage of the host cell. Again, it will give you a little little memory markers, as in how does an antibody actually connects to a COVID virus. I would require 2 more volunteers if possible, and if you would wear gloves, so and just alternate hands, but no, just one glove on one hand, and then the other volunteer would have to. Just wearing gloves because it's and opposite hands, please. So red, yeah. Right on the left one, and then if Dr. Dolan, and if you would come this one, then okay, no, is it the other way? Is it the other? Yeah. It is the other way. So I would just do this because so that my dark color doesn't actually wash off in your bright hands. If it's possible, Dr. Dolan, can we have one of the sides? No, just one of the sides here, that I can just hold so. These are the 2 antibodies, or the 2 antibody bodyguards, who are actually just stopping me from trying to actually get to the SARS-CoV, the SARS-CoV from the host cell. So what I'm trying to do is, whenever I'm trying to be this creepy little guy, sorry, these two antibody bodyguards are actually just hold me. Even if I try to go, they'll actually pull me away, and that is what these two parts. Thank you, lovely ladies. Thank you so much. This is exactly what these 2 parts are trying to do. So the receptor binding protein will be attached with these 2 parts, which are actually, I think it should have been mentioned somewhere. I think it is, yes. So it is the Regen 10933 and then Regen 10987, which stop the SARS-CoV from actually becoming creepy. That's the part. The next part is this. I would also actually encourage you to actually just look at the supplementary data. It has way more data than the original paper. But anyway, so let's look, I will draw your attention to this paper, or this part of the figure actually, where you see there are different regen models present, and this is like a correlation matrix, where this diagonal is self-competition. So the first of each actually is the same. And then there is partial competition, which is yellow, bidirectional competition, which is bad, in red, and then no competition, which is good, okay, for this white part. Now, why did we actually pick? So if you look at the numbers, actually, Regen 1987 and 10987 and 10933 actually have a 0.76 instead of actually like some others having 1.52. So why a smaller number that was picked? Because we want them to be non-competing, as in two distinct parts, which is the left and the right hands, instead of actually both trying to hold. So I can be like a sneaky guy and actually try change my domain and still try and like, you know, grab the host cell. But at least one of them will actually be able to still grab me and hence rendering me still useless. So that part, my ACB link is actually immunology, and that pretty much concludes my presentation about RegenCOV. Thank you.